We just combined the master mix, water, and single-cell suspension. Now we will immediately load the chip.
We have the combined master mix and single-cell suspension on ice. The NextGem single-cell 5'HT V2 gel beads have been equilibrated to room temperature for at least 30 minutes. And we have partitioning oil.
We are ready to load the chip.
For gem generation, load the indicated reagents only in the specified rows, starting from the row labeled 1, followed by rows labeled 2A and 2B, and rows labeled 3A and 3B.
First, we will load the gel beads.
Snap the tube strip holder with the gel bead strip into a 10x vortex adapter. Vortex for 30 seconds. Centrifuge the gel bead strip for approximately 5 seconds. Confirm there are no bubbles at the bottom of the tubes and the liquid levels are even. Place the gel bead strip back in the holder and secure the holder lid. Puncture the foil seal of the gel bead tubes. Slowly aspirate 130 microliters of gel beads. Dispense into the wells in the row labeled 1 without introducing air bubbles. Wait 30 seconds.
Next, we will load the master mix and single-cell suspension.
First, we will process 5 samples. We gently pipette mix the master mix and cell suspension using a multi-channel pipette. Using the same pipette tips, we dispense 140 microliters into the bottom center of wells in the row labeled 2A. When loading the chip, raising and depressing the pipette plunger should each take approximately 5 seconds. When dispensing, raise the pipette tips at the same rate as the liquid is rising, keeping the tips slightly submerged. Next, we will process the remaining 5 samples. We gently pipette mix the master mix and cell suspension using a multi-channel pipette. Using the same pipette tips, we dispense 140 microliters into the bottom center of the wells in the row labeled 2B. Wait 30 seconds.
Finally, we will add the partitioning oil.
Dispense 140 microliters of partitioning oil into the wells in the rows labeled 3A and 3B from a reagent reservoir. Failure to add partitioning oil to the rows labeled 3A and 3B will prevent gem generation and can damage the Chromium X. Close the lid with the gasket already attached. Run the chip in the Chromium X immediately after loading the partitioning oil. Keep the assembly horizontal to avoid wetting the gasket with partitioning oil.
We loaded the reagents into the chip. Next, we will load the chip into the Chromium X.